side of your cheek, you get something material, right? If you see under the microscope, either the onion peel or the scrape of your cheek, if you put iodine or methylene blue or saffron stain to it, if you put iodine or methylene blue or saffron stain to a peel of onion or human cheek cell scrape, all of their chemical components for different regions will be colored differently after taking these stains. When you observe these under a microscope, you see a central nucleus in all these cells. This can be observed only in a microscope. Then nucleus is a double layered structure. It covers the nuclear membrane. Nucleus is a double layered structure, covers the nuclear membrane. Then this nuclear membrane has pores. And these pores allows the transfer of materials inside and outside of the cell. The nuclear membrane contains pores and allows the transfer of materials inside and outside of the cell. And this nucleus contains chromosomes. There is something called chromosome, distinctive feature. Nucleus contains chromosomes. What are chromosomes and what are the functions of chromosomes? Chromosomes are rod-shaped structures. They are made up of DNA and proteins. So chromosomes are rod-shaped structures made of DNA and proteins. DNA here is deoxyribonucleic acid. So then chromosomes contain information for inheritance of features from parents to next generation in the form of DNA. So this DNA determines what characteristics will come from parents to children. So chromosomes are rod-shaped structures made up of DNA and proteins, and this DNA helps in determining what characteristics will be inherited from parent to next generation or parent to progeny in the form of DNA. Then this DNA, many segments of DNA together are called as genes. These genes only tell you what characters you have got from your parents or grandparents, what all you have acquired. Then in non-dividing cells, DNA is present as chromatin material only in non-dividing cells. But when they reach the reproductive stage, again they also form as chromosomes. This chromatin material get converted into chromosomes. Then this nucleus has a role in cellular reproduction, definitely, right? Then these nuclear cells determine how cells will develop and perform and how they exhibit and develop at maturity. So this nucleus determines how cells develop, what forms they take while so this nucleus determines how cells develop, in what way they will develop and what forms they exhibit at maturity, how they look at the time of maturity, and they are responsible for cellular reproduction. And in bacteria, there is no nuclear membrane. So in other plant cells there is nuclear membrane, but in bacteria there is no nuclear membrane. Only nucleic acid is there. This nucleic acid is called as nucleoid. So in bacteria there is no nuclear membrane. Only nucleic acid is there. This nucleic acid is known as nucleoid. Then talking about prokaryotes and eukaryotes. So prokaryotes are present in only lower living organisms and eukaryotes are present in higher level of organisms. So these prokaryotes do not have nuclear membrane, whereas these eukaryotes have this nuclear membrane. Both of them have nucleus, but they do not have nuclear membrane, they have nuclear membrane. Prokaryotic cells are smaller in size. Eukaryotic cells are larger in size. Then prokaryotes lack the cell organelles. Eukaryotes have the cell organelles. Then prokaryotes have one chromosome. Then eukaryotes may have more than one chromosome or many chromosomes. Then prokaryotes. Photosynthesis in prokaryotes is carried out by a process only with vesicles. Whereas in eukaryotes, the photosynthetic activity is associated with plastids. Plastids is cell organelle of the cytoplasm. So this is about nucleus. So the last part of the cell we will be going to deal with cytoplasm. Cytoplasm is a fluid content inside the plasma membrane. So when we stain any cell, like an onion peel cell or the scrape of the human cheek with methylene blue, saffron or iodine, what we see under a microscope, we see a lightly stained area. That is the fluid content of the cell. That is only cytoplasm. So it can be seen as the lightly stained area in the onion peak of human cheek cells only under the microscope. So all the cell parts and the cell organelles can be seen only under the microscope. We cannot see them with naked eye. So this is the fluid content inside the plasma membrane. Then it contains many organelles. They are enclosed by membranes. So example, virus. So virus, when living separately, do not show any characteristics. But when they enter the human life, human body, they then Example, viruses. Viruses shows characteristics of life only after they enter the living body, as they lack these cell membranes, as they lack this cytoplasm. So that is the reason virus shows the characteristics only after entering any living body, because they lack cell membranes or cytoplasms. Then in prokaryotic cells, there are no membrane bound cell organelles, whereas in eukaryotic cells there are many cell bound organelles. So in detail we should know about these cell organelles. Cell organelles are 6 types. They are present inside the cytoplasm. So normally cells need lot of chemical activity to support the function. That cells make up to the organs of the body, then organs make up the organ systems make up the organism. So there is lot of chemical activity going on to support the structure and function of the life. So to keep these all the activities separate from each other, there are some substances called cell organelles. They are of 6 types. They are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. Now we shall be dealing in detail with these 6 cell organelles, and these are very important. They carry out the crucial functions of the cell. So the first cell organelle we are going to deal is endoplasmic reticulum. Endoplasmic reticulum forms a large network of membrane bound tubes and sheets or vesicles or bags. So endoplasmic reticulum is a large network of membrane bound tubes, sheets, bags or vesicles. Then the structure of endoplasmic reticulum is similar to that of plasma membrane. Then there are 2 types of endoplasmic reticulum: rough endoplasmic reticulum RER or smooth endoplasmic reticulum SER. Rough endoplasmic reticulum is rough. There are ribosomes attached to it. Whereas smooth endoplasmic reticulum, it helps in manufacture of fats and lipids. Then these fats and lipids are used for all the cellular functions. Then also smooth endoplasmic reticulum helps in building of the cell membrane. So this process is called as membrane biogenesis. The smooth endoplasmic reticulum helps in building of the cell membrane. This process is called as membrane biogenesis. Then some lipids and fats sometimes acts as enzymes and hormones and help in the cellular processes. One more main thing you have to remember is, it may be asked as a fill in the blank objective or a small question. This endoplasmic reticulum, in which separately smooth endoplasmic reticulum in the liver cells of vertebrates or animals, the function of these smooth endoplasmic reticulum is detoxification of poison and drugs. In the liver cells of vertebrates or animals, smooth endoplasmic reticulum function is detoxification of poisoning and drugs. So this is about endoplasmic reticulum.